So last time when we were together, we were talking about how the structure of the amino acid side chain influences the conformations that can be adopted by a particular amino acid. The example that we used was alanine, which in the Ramachandran plot to the right, we see that alanine is comfortable in an alpha helical area. Again, the only reason we know that area is associated with alpha helices is we've looked at alpha helices, we've measured the dihedral angles, and that's what they are. And so alanine's pretty comfortable there. A little bit less comfortable in a beta sheet conformation. Again, we know that this is beta because we've seen beta sheets, measured the dihedral angles, and this is sort of the area they show up in. I showed you last time how adding two branching methyl groups to the beta carbon of alanine to give you valine increases the population up in the beta region of the plot. That tells you something about how side chain structure influences conformation. Presumably, something about having branching at the beta carbon makes it more favorable to adopt these types of conformations. We showed you another example of that in the case of serine, shown here, which is like alanine but with an OH group. Like alanine, serine's pretty comfortable in an alpha helix shape, a little less comfortable in a beta sheet or beta strand shape. But you put a branching methyl group on the beta carbon to get threonine, and suddenly the population up in the beta region is much higher. So again, side chain structure changes the steric environment around the phi and psi angles and changes which conformations are more favorable there. The other example of side chain structure influencing dihedral angles was just to notice that in most of these Ramachandran plots, the positive values of phi are not really highly populated, presumably because there's steric clashes over here when phi gets to a positive angle. Not so for glycine, because glycine lacks a side chain, and so areas in the positive region of the Ramachandran plot are now accessible. There's steric clashes that don't happen for glycine that do happen for the other amino acids. Another example of side chain structure and shape is to consider alanine, where, as we've said before, phi is restricted to negative values, but psi can be both positive and negative. And phi ranges from about minus 60 to around minus 180. If you look at proline, which is kind of weird because in proline, the beta carbon is tied back onto the amide nitrogen in this 5-membered ring. What that does is that restricts the values that psi can adopt to an even narrower range. Notice that proline only adopts psi values near minus 60 degrees, can't get to minus 180. The reason it can't get to minus 180, you can see if you build a model of a 5-membered ring and imagine trying to twist it around like this without breaking the ring, and you can't. So the presence of that ring really does restrict the values that phi can adopt. And so proline will do different things than some of the other amino acids because of that constraint. Okay, so relationship between side chain structure and function. You've seen one example of that. Side chain structure changes the dihedral angles that an amino acid can adopt. What we're going to do today is look at the actual structure of the side chain, learn some things about them, and what kinds of non-covalent interactions they can participate in. So I will go through each of the 20 amino acids that are shown here. These are the 20 that are encoded for in DNA and RNA. There are some other amino acids that are important in biology but aren't coded for. We won't talk about those. You can learn about those as you progress in science if you're interested. So I've grouped these 20 amino acids into categories that make sense to me. You don't have to group them in categories. Some of them don't fit very neatly into specific categories. They sort of straddle a couple, but we'll just sort of go through them in this order. We'll start with non-polar amino acids. You need to know their names, structures, one-letter codes, and side chain pKa's as appropriate. Before we dive into that, I just want to say that there's a table in your text that shows pKa's of the amino acids, you may be somewhat overwhelmed by that table, thinking, do I have to memorize all of that? And the answer is no, you don't. Let me show you how. This is the amino acid aspartic acid. Your text shows 3 pKa's for this amino acid. One is for the backbone carboxylic acid group, which has a pKa of 2 point something. The other is for the backbone ammonium group, which has a pKa of 9, and then the other is for the side chain group, which has a pKa of around 4. Now, there are, and different amino acids will have slightly different backbone pKa's. You'll learn that, for glutamic acid it's 2 point something, and for aspartic acid it's 2 point something else. That kind of distinction doesn't matter. So you don't need to memorize the backbone carboxylate and ammonium pKa's for each individual amino acid, because they're all basically the same. It's enough to know that the backbone is 2 and the ammonium is around 9. The other reason we don't worry too much about backbone, about pKa's of backbone groups is because in a larger peptide or protein, like I think I illustrated for you last time, most amino acids aren't going to be at the N-terminus or the C-terminus. So most amino acids in a peptide or protein aren't going to have a charged backbone group. Most amino acids are going to be in this internal environment where it's an amide on the right and an amide on the left. Only the C-terminal amino acid will have a free carboxylate and it'll be negatively charged. Only the N-terminal amino acid will have a free ammonium group and be positively charged. So for the most part, you should focus on pKa's of side chains. That said, this first group of nonpolar amino acids doesn't have any side chain pKa's to know. So this is alanine. The one-letter code makes sense. Alanine's just a methyl group. Valine is like alanine, only at the beta carbon we've attached 2 methyl groups. This is the 2 branching methyl groups. These 3 carbons, beta carbon and then the 2 gamma methyl groups, could be called an isopropyl group. It's sort of easy to remember because it looks like a V if you look at it from the right angle. So valine side chain looks like a V. It's beta branched, as we've talked about. Leucine is like valine. You just insert a CH2 unit in between the isopropyl group and the alpha carbon. So in leucine, the beta carbon is CH2. Now it's the gamma carbon that has the branching methyl groups on it. And then isoleucine is like leucine, only you've taken one of the methyl groups and you've moved it over back to the beta carbon. That's why it's called isoleucine, because it's an isomer of leucine. The one-letter codes make sense, L and I respectively. One of the tricks with isoleucine is once you move that methyl group back, suddenly the beta carbon is a stereocenter. And it turns out natural isoleucine has all one configuration at that beta carbon, and it's S. Now, people get that mixed up and confused all the time. If you draw the other stereoisomer, it's not actually isoleucine anymore, it's a diastereomer. Nature uses isoleucine, so, but I'm not going to require you to know the configuration of that atom. In part because on a good day, if I'm not paying attention, I probably don't draw it right myself. So as long as you've got the carbon chain for isoleucine, you know there's branching methyl group of the beta carbon. You don't need to know the stereochemical configuration of that beta carbon. Of course, you do need to know the stereochemical configuration of the alpha carbon, which is the same for all the amino acids. All right, then lastly, you've got methionine. Methionine's nonpolar. It's a linear chain that goes out from the alpha carbon 1, 2, 3, 4 atoms, beta, gamma, delta, epsilon. Only instead of the delta atom being a CH2 group, it's a sulfur. Sulfur's is not particularly electronegative. So methionine fits in as a description of, it fits in with the other nonpolar amino acids. Though the sulfur can get oxidized in an oxygen-rich environment to form this sulfoxide, and that will make it more nonpolar. This happens all the time in my lab. This is important in some cases in biology. For you, just thinking about the methionine side chain, this thioether functional group being nonpolar is good enough. So that group of nonpolar amino acids is relatively simple. Nonpolar groups don't interact particularly well with water. They prefer to be sequestered away from water for reasons that we'll talk about a little bit later. Okay, the next group of side chains are also mostly nonpolar. We're gonna put them in their own group because they are all aromatic. We'll start with phenylalanine. At this stage, the PH sound makes an F sound, so using the one-letter code F sort of makes sense. So F is for phenylalanine. It's got a benzene ring attached to the beta carbon. And you're used to thinking of benzene as being pretty nonpolar. We're going to come back to this as we look at non-covalent interactions among side chains, but there's more going on with a benzene group than you think. And the reason for that is that sp2 carbons are more electronegative than hydrogens, meaning that all of the hydrogens around the benzene ring should be electron poor, and all of the carbons should be electron rich. All of those individual bond dipoles cancel each other out. There's not a really strong net dipole for a benzene ring, but there is something called a quadrupole. Quadrupole refers to something that isn't a dipole but is also still not uniform electron density distribution. We mean that the face of the ring is electron rich, the edge is electron poor. That's going to have some consequences for the kinds of things that aromatic side chains can do. Tyrosine is like phenylalanine, only we've taken the para carbon and we've put an OH group on it. We use the one-letter code Y because we're going to use T for something else, and this is BYU's favorite amino acid. Yeah, whatever. Yeah. Grown, go, yeah, so. I've got some dumb jokes that are going to help you remember things, so we'll just continue on with them. Tyrosine's OH group can be a good hydrogen bond donor. It also has a pKa that I want you to know. The pKa for this phenol OH group is around 10. Tyrosine can be nucleophilic. Sometimes it can get deprotonated and act as a nucleophile. You'll see in biochemistry that sometimes the oxygen of tyrosine attacks a phosphorus and you get a phosphorylated version of tyrosine, which is important in lots of cell signaling events. We still consider tyrosine to be mostly nonpolar, even though it's got this polar OH group. And its similarity is that it's aromatic. Like phenylalanine, the face of the tyrosine ring is electron rich. The edge is electron poor. The next aromatic amino acid is tryptophan. This one we sort of go off the rails and just pick a letter from the alphabet because we're using the T and the Y for other things. So W. If it's 1930s and you are wearing a hunter cap with a shotgun and you're hunting a rabbit, you might, and your name is Elmer Fudd and you're on Looney Tunes, you might pronounce tryptophan as twiptophan, which may help you remember the W. I don't know. But there you go. And tryptophan is different than tyrosine or phenylalanine. It's a bigger aromatic ring. You do have a benzene ring, but then you also have this 5-membered ring containing the nitrogen and a double bond. This is aromatic. You can count the pi electrons, 2, 4, 6, 8, and then the lone pair counts. Why? Because it's in a p orbital adjacent to a pi bond, so it's going to be in a p orbital to participate in resonance. 10 electrons, definitely aromatic. That type of fused ring of a 6-membered ring and a 5-membered ring in this way is called an indole, and the NH is a hydrogen bond donor. We're not going to worry about its pKa. It is high enough that we don't worry about it in biological or physiological pHs. Like tryptophan and like phenylalanine and tyrosine, the face of the tryptophan ring is electron rich. The edge is electron poor. Now the next is histidine, and now we're back in a world that makes sense because its one-letter code is H. Histidine side chain is aromatic, but it's also more polar than the other aromatic side chains. So it doesn't necessarily fit neatly with the other aromatic side chains. I grouped it here with other side chains that are normally positively charged at physiological pH called cationic. Histidine doesn't neatly fit in with those either because histidine's pKa is such that at physiological pH it's mostly neutral with about 10% positive charge. So this is what I meant. Histidine straddles a couple of different categories. It doesn't fit neatly into either one. Here is the neutral form of histidine called imidazole. You can tell that it's aromatic because 2, 4, and then 6 electrons involved in the pi system. You can tell that the lone pair on that nitrogen is in a p orbital because it's adjacent to a pi bond, and so it's going to participate in resonance. In contrast, this lone pair is in an sp2 orbital. It has to be because the p orbital from that nitrogen is already involved in that pi bond. So the imidazole is aromatic. If it picks up a proton on the sp2 hybridized lone pair, then it becomes this cationic imidazolium ion, which is still aromatic. You should be able to see how that positive charge is stabilized by resonance and could be on the other nitrogen as well if we move this lone pair over to form a pi bond and then move that pi bond over to be a lone pair on the nitrogen. The pKa of that positively charged imidazolium ion is 6, and that's a number you need to know. At pH 7, you would predict, that's one unit, one pKa unit is a factor of 10 in equilibrium constant. So you would predict about a 10-to-1 ratio of neutral to positively charged. So at physiological pH, you're going to make the judgment call that histidine is neutral because it's majority neutral. But you should also not be surprised to see histidine in its positively charged imidazolium form in some enzyme catalyzed reaction mechanisms. Basically, you expect histidine to have access to both forms. It can act as a base to remove a proton. It can act as an acid to give a proton. And histidine's found because of this in a lot of enzyme active sites. So don't be deceived. I grouped it with the cationic ones because it can be cationic about 10% at physiological pH. It's mostly neutral at physiological pH. It could also fit in with the other aromatic amino acids. Okay, questions about that? So the other positively charged ones, lysine. The big challenge here is remembering how many carbons are in the side chain. I always remember the nitrogen in the lysine side chain is on the epsilon carbon, and then I memorize my Greek letters, so I'm not sure that helps anybody. Alpha, beta, gamma, delta, epsilon, then the ammonium group. At physiological pH, that is almost always charged. pKa of the ammonium is 11. That's another number to know. Of course, it'll also be in a table of values at the end of the exam. But I found that to answer questions quickly on an exam about amino acids, it's good to just know. So lysine is almost always positively charged at physiological pH. At pH 7 with a pK of 11, this should be plus. Lysine can exist in its neutral form and can sometimes be a nucleophile, but again, if you see lysine, you're going to think positive charge. Its one-letter code is K, inexplicably. And I've known that for like 25 years, and so it's second nature to me now. I think lysine and I think the letter K. You're not there yet because you just learned it. It doesn't make sense. You're going to have to figure out a way to memorize it. The next amino acid is arginine. Arginine is also always positively charged. Arginine is different from lysine because it's sort of one carbon shorter. The nitrogen is on the delta carbon for arginine. So you just go alpha, beta, gamma, delta, and then a nitrogen. The group here is one functional group. It looks like 3 nitrogens, but it's one single functional group called a guanidinium group. The positively charged nitrogen, the positive charge on that nitrogen is delocalizable via resonance on the other 2 nitrogens. To see that, simply push the lone pair here to form a pi bond, then push the pi bond here out onto the nitrogen to form a lone pair. And you can see how that positive charge can be on each of those 3 nitrogens. So this is one functional group, positively charged with a pKa of 12. It's called a guanidinium group. When it loses its proton, it forms this neutral guanidine, but arginine is always, because the pKa is 12 at physiological pH, it's always positively charged. The R for the one-letter code sort of makes sense because the second letter of arginine is R, and it's also the pirate's favorite amino acid. R, right? Ha ha ha, there you go. Yeah, thank you. That was all it was good for. We'll rate that one 2 out of 10. Okay, so lysine and arginine, always positively charged. Histidine, sometimes, but at pH 7, you're going to call it neutral. Does that make sense? All right. So the next are the negatively charged ones. These I've named according to their negatively charged conjugate bases, which is the major form at physiological pH. Aspartate and glutamate. Here we've given up on letters that make sense. It's D for aspartate, it's E for glutamate. You're just going to have to figure out a way to memorize that. For aspartate, in its acid form, which has a pKa of 4, you've got a carboxylic acid on the gamma carbon and just a beta carbon, and beta then gamma, and the gamma is the carboxylic acid. Glutamate is 1 CH2 unit longer, so you go alpha, beta, gamma, and then delta is the carboxylic acid. But as I said, at physiological pH, these are present in their negatively charged forms. So that's why I call them aspartate and glutamate. One interesting thing to observe here in the Ramachandran plot is that apparently having the carboxylic acid group or carboxylate group closer to the backbone opens up some conformations that aren't available to glutamate. Notice that aspartate can adopt some positive values for phi that a lot of the other amino acids can't. It's possible that it's hydrogen bonding between carbonyl and OH and maybe some backbone atoms that make those conformations possible. But you'll notice just adding the CH2 unit in the backbone then gets rid of that. So that's kind of an interesting feature of aspartate. But when you see D or E, you need to be thinking negative charge. And those are pKa numbers to know. Next, we've got polar amino acids that are neutral, so not usually negatively charged at physiological pH, but still polar, capable of hydrogen bonding. Serine you've seen before. This just has an OH group on the beta carbon. And then threonine has a branching methyl group on the beta carbon. Threonine's beta carbon is also a stereocenter. Again, I'm not going to care if you don't remember what the configuration of that beta carbon stereocenter is. Threonine and serine both have the potential to be nucleophiles if they're deprotonated. Their pKa's are both 16. That may or may not be in the table in your text, but you've learned already in 351 that alcohols have a pKa of about 16. You'll see serine as a nucleophile in some protein active sites, but it's got to get deprotonated first. And so there are other side chains that do that deprotonation. All right, serine, threonine, the one-letter codes make sense. This is cysteine. And cysteine is like the sulfur version of threonine. Instead of an OH, you've got an SH. And I just learned this one in this dad joke in between the classes, so I'm trying it out here for the first time. You can remember that cysteine, you can remember what cysteine is because it's got an SH, and you should be quiet when you're in the Sistine Chapel. Oh, that's a slapper, isn't it? Yeah. All right, whatever. The changing from serine to cysteine, because sulfur is so much bigger than oxygen, the conjugate base of cysteine is more stable than the conjugate base of serine because it spreads out electron density over a larger region of space. Therefore, the pKa of the cysteine side chain, SH, is lower. It's about 8. So at physiological pH, you expect it to be neutral, but you can realize that you can have access to the negatively charged version, which is actually a really good nucleophile. And you will see cysteine act as a nucleophile in biochemistry. Another interesting thing cysteine can do that other amino acids can't is the sulfur is oxidizable, and you can form what's called a disulfide, in which two cysteine side chains get together. They lose basically hydrogen. They lose H plus and H minus, so one equivalent of H2. They lose it to form this cross-link between sulfurs. That's called a disulfide bond, and that is an interesting thing that imparts structural stability in secreted proteins, especially extracellular proteins. Peptide hormones like insulin have disulfides. Cell surface receptors, antibodies have disulfides. Your hair, keratin, has disulfides in it. In fact, back in the 80s when this kind of thing was popular, if you wanted curly hair, you could go to the salon and get a perm. I don't know whether they still do this or not. But what they do is they reduce the disulfide bonds in your hair with a chemical. It's going to smell a little bit like sulfur because that's one of the reducing agents for molecules that have sulfur in them can reduce disulfide bonds. And then they wrap your hair around whatever, put it into a curly shape, and then they reoxidize all the disulfide bonds with an oxidizing agent, and those disulfide bonds hold the hair in place. So kind of interesting that works. They're doing disulfide bond chemistry on you if you go do that. There are two other neutral but polar amino acids, and these are going to be cousins to aspartic acid and glutamic acid. These are the amide versions of aspartic acid and glutamic acid. So aspartic acid has an OH group on the delta carbon. The asparagine is going to have an NH2 group on the delta carbon. I said that wrong. I got to go back. I'm sorry. Aspartic acid has a carboxylic acid group as the gamma carbon, an OH group, and a carbonyl. In contrast, glutamic acid still has the carbonyl, but now has an NH2 group. So this is the amide version. Biologically, nature makes asparagine from aspartic acid, so it makes sense that their names should be similar. The one-letter code is N, and again, we've just given up. We're running out of letters, so we got to pick something, and N is the right letter. There's an N in asparagine, so whatever. Glutamine has a CH2 unit added to the side chain relative to asparagine. Its one-letter code is Q. Interestingly, as with aspartic acid, you see asparagine adopt some phi values in the positive region. Again, that's probably because of these hydrogen bonding groups and their proximity to the backbone. And then lastly, you've got glycine and proline. Thankfully, these one-letter codes make sense. These are the weird ones. We've talked about glycine's flexibility. We've talked about proline's restricted phi angles because of the cycle. One other thing to know about proline is it tends not to be present in helices because helices involve hydrogen bonding with amide protons, and proline doesn't have one. Because the side chain takes the place of the proton that's normally on the proline. Proline can't participate in hydrogen bonding in a helix. You'll see that glycine and proline are frequently found in places where secondary structure changes. So if in a protein a helix needs to stop, have a turn, and then form another helix, often glycine or proline will be in those turns. Similarly with a beta sheet, if a strand is going along and then needs to turn and then go back, often glycine and or proline will be in those turn regions. Okay, any questions about the 20 basic amino acids that we've talked about? Yes?
Which ones do we, which side chains do we have to worry about stereochemistry?
You don't have to worry about side chain stereochemistry. Yep. You do need to know names, one-letter codes, structures, and side chain pKa's as appropriate. And then, given knowing what you know about the side chains, you ought to be able to have some idea of how their properties differ. We're going to talk about that next as we talk about the kinds of non-covalent interactions that these side chains can participate in. Other questions? Yeah?
So do we need to know the essential amino acids?
Do you need to know the essential ones? Yeah, I put that in the study guide once because I thought it was important. If you are in nutrition and dietetics, you probably already know it. The fact is, off the top of my head, I don't know it, so I'm going to call it audible and say don't bother unless you really want to know. Essential amino acids are the ones that our biology doesn't make, so you have to get it from your diet, from plants and other animals. So it's kind of interesting that as organisms we evolved to stop making some of the essential amino acids because it was just easier to consume other life to get it. But we are nothing if not opportunists as humans, so yay us. Another thing, I guess, I don't know a ton about this. I want you to be sensible consumers. I know that the entire forces of social media are constantly marketing products to you with promises that if, like Lightning McQueen says in Cars, with Rusty's medicated bumper ointment and an insane amount of luck, you too can look like me, right? People are trying to market products to you. You'll see people advertising protein powders with BCAAs as though you're supposed to be excited by that. What are BCAAs? Those are branched chain amino acids like valine and threonine and isoleucine. I'm not sure why they're telling you that that's awesome and trying to market to you that you should And so somebody that knows about nutrition can tell me whether that's important or good or not, but that's what they're talking about. Go ahead.
I mean, along that same vein, I've seen in a lot of protein powder type that have those added essential amino acids. Just this is how, I guess, do those impact your workout? Like, do they impact like muscle building?
Yeah, so that's a question that I am totally unqualified to answer. I will say that I don't know. Protein powders, they got to get the protein from some source. As to whether it exists as intact protein or chopped up amino acids, I'm not sure. Your body, when you eat protein, digests the protein into chopped up amino acids, and then that's absorbed through your stomach, and that's all the biology I know. I'm so sorry. A lot of, for example, protein powders are from whey, which is milk, so they've got casein and some other milk proteins in them. Ideally, if you want to build muscle, you need to give your body the tools to do that. Muscle is mostly protein, so you need amino acids to do that. As to how much you need and whether you need, the essential ones are the ones that your body can't make, so you have to get them. So it's good. As to whether you need to be supplementing or you can get it through your diet by eating meat and other sources of protein, I don't know. That's the department over in food science, nutrition, dietetics, and they can tell you more. Yeah. Okay, cool. But yeah, just be thoughtful consumers about information. Realize that anybody on social media is trying to sell you something. Go ahead.
For the find site from a Contron,
Yeah.
you said that we don't have to memorize those, but just be able to tell what.
Right, you don't need to memorize it, but given data, can you draw some conclusions? And I've walked you through some examples of that. Which is more comfortable in a beta sheet conformation, alanine or valine? Given data, could you tell? And I need to amend my statement. Not everybody on social media is selling you something, right? So there are people out there doing good things. But yeah, just be thoughtful consumers of information. All right. Sorry, painting too broad. I need to stick to the science and avoid the social commentary. All right, so let's talk non-covalent interactions in peptides and proteins. This is key to having an idea based on the relationship between sequence and structure and function. Having an idea when you see a one letter in an amino acid sequence, what that side chain looks like, what it might be able to do. So this is going to be a review of things that you learned in 351. We'll go through it pretty quickly. Non-covalent interactions are generally interactions that involve either full or partial charges. They're generally worth about 1 to 10 kilocalories per mole, which is about a factor of 10 or 100, between 10 to 100 times weaker than, say, a carbon-hydrogen bond. Here are some that you will see that are pretty common. So one is called a salt bridge. In 351, you would have called that an electrostatic interaction. Here's an example, a lysine side chain, alpha, beta, gamma, delta, epsilon, and then the ammonium group, positively charged nitrogen, and then nearby there might be a negatively charged, say, glutamic acid, E, in its negatively charged carboxylate form, and the electron-poor ammonium is near the electron-rich carboxylate. You have a complementary charge interaction. Positive charge attracts negative charge. It's essentially a Coulomb's law thing. It'll be stronger if the two groups are closer to each other, right? These often show up. And so any of the positively charged amino acids, say K or R, and then any of the negatively charged amino acids, say D or E, can participate in salt bridges. So when you see a K or an E or an R or a D, you should be thinking, okay, there's going to be a charge, and that might interact with a complementary charge. Another type of interaction that you should be aware of is hydrogen bonding. We can't possibly illustrate all the examples. This hydrogen bonding can occur between backbone groups. It can involve backbone groups, it can involve side chain groups, and it can definitely involve water. And so I'm just going to show you an example of a hydrogen bond between backbone groups in a peptide. Here is a carbonyl oxygen from one amino acid. And then here is going to be a nitrogen of another amino acid. And the carbonyl of another amino acid. So you can see things. I'm showing you actually two such hydrogen bonds. This is a kind of hydrogen bond that occurs between beta strands in a beta sheet. And essentially it's a dipole type interaction. You have the positive end of a dipole associated with the NH bond interacting with the negative end of the dipole associated with the CO bond, or alternatively a partial negative charge on the oxygen and a partial positive charge on the hydrogen. These hydrogen bonds can be quite strong. Again, they require proximity. They're stronger if the atom that's negative is closer to the hydrogen that's partial positive. And these can occur, again, between groups in the peptide backbone, between groups in the side chains, between water, and all possible combinations of those. So these occur all over the place. They're important. There's a particular hydrogen bonding pattern that's present in an alpha helix that we'll talk about in a little bit. Here's an unusual one that you probably didn't learn about, maybe didn't learn about in 351. We're going to call these pi interactions. And these are based on the idea that a benzene ring, though it's nonpolar, has a strong quadrupole, which means that because the sp2 carbons of the ring are more electronegative than the hydrogens, that you expect the face of the benzene ring to be electron rich, and then you expect the edge of the ring to be electron poor. And this means that benzene rings can interact with charges or partial charges in ways that maybe you didn't expect. So, for example, imagine this phenylalanine side chain. Here's the alpha carbon. And I'm going to go ahead and draw in the hydrogens. We're going to use this phenylalanine side chain multiple times, so I'm going to quick draw it and illustrate it, and then if you'll forgive me, we're going to do a little bit of copy and paste. Imagine that the electron-rich face of the benzene ring is interacting with the electron-poor ammonium group, the positively charged ammonium group of a lysine side chain. So we'll color this lysine side chain ammonium group red for electron poor. Imagine that nitrogen moving down to be close to the benzene ring. The positively charged nitrogen would be attracted to the electron-rich face of the benzene ring. I'm going to illustrate that with the red dotted line. You got the idea. And maybe just for the sake of the drawing, I'm going to erase the red electron-rich portion, the red electron-poor edge, just so it can be clear that that positively charged nitrogen can get quite close to the aromatic ring. That's called a cation pi interaction, and those are found all over the place in proteins. With different cations as well. Sometimes you see it with an arginine side chain, sometimes you see it with potassium or sodium ion, and sometimes you see it with other types of nitrogen positive charges. Next, you could imagine that you could bring two aromatic side chains together, but you wouldn't expect them to be able to stack on top of each other in a straight line, because then the negative electron-rich faces would repel each other, the positive electron-poor edges would repel each other. So what you tend to see is something like this, which we would call an offset. We would call this offset pi stacking. And in offset pi stacking, the rings are sort of parallel to each other but shifted so that the electron-rich face of one hovers over the electron-poor edge of the other. You tend to see this. The other format that pi interactions can take would be for the electron-poor edge of one benzene ring to sort of point downward towards the electron-rich face of another benzene ring in this kind of orientation. Sort of a T-shaped, oops, I didn't circle the whole thing, sort of a T-shaped interaction. So we call that sort of T-shaped pi stacking. All of those things can happen in with benzene rings. And then finally, the most important one that we need to mention is the hydrophobic effect, which is not the same as van der Waals interactions. Van der Waals interactions are relatively weak. To illustrate this hydrophobic effect, we're going to consider the unfolded versus folded conformations of a peptide or a protein. We're going to do this in cartoon form. So there's an unfolded protein, and then here is a folded protein. In the unfolded protein, we're going to have a few nonpolar side chains, a valine, a leucine, perhaps another valine, and then we'll have some polar side chains, maybe a positively charged one, maybe a negatively charged one. The positive and negative charges do just fine interacting with water, right? The negative end of the water dipole can interact with a positive charge. The positive end of the water dipole can interact with a negative charge, sort of via hydrogen bonding. All of that's okay. Those groups are pretty fine in water. But if we zoom in around a nonpolar side chain, the nonpolar side chain lacks the functional groups to, say, hydrogen bond with water. And therefore, water molecules that are closest to the nonpolar side chain have space nearby where they would normally have been hydrogen bonding with water, but they can't because there's not water there. It's the nonpolar side chain. So what that causes is that the water molecules in the immediate vicinity of the nonpolar group start to slow down, and they tighten up their interactions with the nearby water molecules. They've lost some interaction, so to compensate, they start interacting even, oh, sorry, somebody was going to tell me I did that. Yeah, yikes is right. Anyway, now I can't talk because can't talk and do this at the same time. Anyway, the water molecules in the immediate vicinity of the nonpolar side chain slow down, increase the strength of their interaction with the remaining water molecules that are close to them, and they form this ice-like cage of water molecules that are moving more slowly and therefore have lower entropy than a water molecule that's either interacting with something via a hydrogen bond or that's just swimming around in solution. And all of these nonpolar side chains have this ice-like cage when exposed to water. When the protein folds, it tends to sequester the nonpolar side chains in the interior of the protein. Some people think this is because the nonpolar side chains are engaging in strong van der Waals interactions with each other. But those van der Waals interactions are weak. What's really happening to drive the folding process is all of these water molecules that used to be constrained and slow and in this ice-like cage around the nonpolar side chains are now zooming around free with much, much increased entropy. And to illustrate this point, one of them is shouting, "I'm free," and the other is shouting, "Wee," and they're just zooming around, okay? Those are zoom arrows. There should be a little cloud of dust, like when Wile E. Coyote or the Roadrunner runs. Anyway, and the positively charged and negatively charged side chains are going to be mostly on the surface of the protein. This is called the hydrophobic effect. It's entropy-based. The water molecules that are now zooming around and were released have more entropy, and therefore this process is spontaneous. The free energy of folding is downhill, but it's downhill mostly because of entropy. If you remember that entropy, the free energy is delta H minus T delta S. Oh, I'm struggling today. I'm trying to hurry and I'm just making all kinds of mistakes. Delta S is entropy. If entropy increases, delta G gets more negative. And so the hydrophobic effect drives protein folding. And so you want, in general, to put nonpolar amino acids in the interior of a protein. We're going to use these principles. Which side chains are charged? Which ones can do salt bridges? Which ones can make hydrogen bonds? Which ones can do pi-type interactions? Which ones are nonpolar and prefer to be buried in the interior? As sort of rules to help us understand how amino acid sequence relates to structure. And we're going to do that with a sequence, which I'm going to write for you here. And then we're going to work our way through it and try to understand what kind of structure the sequence directs to have happen. So I'm going to write it down, a series of one-letter codes. You need to understand to begin with that peptide sequences are always, always, always written from left to right, C terminus to N terminus. Never, ever C to N. That's important, you need to know it, and I will trick you on that in the future if you're not careful. So that is your warning. Are you scared? Am I evil? Yes. So just beware, N to C. The first amino acid in this sequence is arginine. It's at the N terminus. That N-terminal amino group would normally be positively charged. In this peptide, it's been modified with an acetyl group to make it an amide. That's not a natural modification. That's something we do in the lab, and I've made this peptide before, and so that's a detail you don't necessarily need to worry about, but I'm putting it here for accuracy. So here is the sequence, RMK, QLEDK, VEEL, SK, N, YHL, EN, E, VARL, KK, LVGER. And now, in the second third of the sequence, I'm just kidding, that's it, okay? This is the C terminus, and the C-terminal arginine has a free carboxylic acid group, okay? So that's the sequence. I think it's about 33 amino acids long. What I want you to do now is, based on what you know about the 20 amino acids, I want you to identify all the amino acids that are positively charged and put a little plus by them. Zoom out. Yep, sorry. All the amino acids that are positively charged and put a little plus by them. All the amino acids that are negatively charged and put a little plus by them. Figure out what the total net charge is on the peptide. I also want you to identify all the amino acids that are in that are nonpolar. And let's say this. I want you to identify any amino acid that is more nonpolar than alanine, and I want you for now to ignore the aromatic amino acids. So basically, you're looking for things like valine, leucine, isoleucine, and methionine. Okay? Yes?
What pH?
And, oh yeah, pH is going to be 7. Okay. All right, we'll see you Friday. Have a good week.